from Environment and Climate Change Canada, and the talk is going to be the Lake Huron lower trophic level changes from 2017 to 2022.
You guys hear me at the back there? Good? Okay. Hopefully don't wreck the slides here. I'm presenting today on behalf of both myself and Annie Schofield. We're the co-chairs of the Cooperative Science and Monitoring Initiative under the Annex 10 of the Great Lakes Water Quality Agreement. None of the work that you will see today is mine. It is a compendium of stuff that has been contributed from various partners, which I will show you what I've got here today. These are the folks who've made it possible for me to talk about something other than just empty slides. If you have questions, they're the ones to contact, and I'm more than happy to put folks in touch with them since they may not all be here, and there are no emails on the screen. For those who are not familiar with the Cooperative Science and Monitoring Initiative, or we have abbreviated as CSMI, it's a 5-year rotational cycle. Effectively, we take priorities that are developed through the lake-wide management plans and then begin to sort of implement project planning phase. The third year is the intensive field year, and that's what the results that I will show today are primarily derived from. And then there's a year of data analysis, and then of course reporting out. So for Lake Huron, the reporting out was actually last October. So it was a little bit behind, but we are now getting ourselves back and re-energized on the 5-year cycle. So every 5 years there is a intensive field year. This year, of course, being Lake Michigan, I have very little involvement in that, but the year after we will be back up in Lake Superior. The priority areas, at least at a high level, cover things from chemical contaminants, nutrient and bacterial pollution. Habitat species is a very broad, encompassing area. Invasives and then other stressors is kind of the bin where everything that doesn't already fit into one of the first 4 bins gets stashed. I haven't done the verbose aspects of the priorities here, but you can find them in each of the lake management plans, which are published by national.net. So I'm going to start with nutrient status, and for Lake Huron, of course, one of the concerns is total phosphorus in terms of productivity at the base of the food web. The blue bar is 2017 data. The orange bar is 2022 data, and suffice it to say, there really hasn't been much of a change. It's still very low when you look at the lake-wide average. There are, of course, spots where it's above these typical 3 to 5 microgram per liter values, but for the most part, it remains around 3 to 3.5 micrograms per liter for total phosphorus. Silica started to increase back in the mid-2000s, and we sort of interpret it as a basis indication of how much diatom growth has occurred. So sampled in spring, there should be lower dissolved silica if you've got lots of diatoms. In this case, we have lots of dissolved silica, which sort of infers fewer diatoms. High level, really no change since 2017, which is perhaps not surprising. I've included this slide, which is a bit more complicated. Karen Binding, who's a remote sensing expert with us at Environment and Climate Change Canada, has spent the last little bit reanalyzing a lot of satellite imagery for Lake Huron, and both in terms of water clarity, so the Secchi disk depth type metric, but also chlorophyll, which is that other indicator of how things are going at the bottom of the food web. And one of the pieces that really sticks out is this bottom chart here. In the blue is the pre-2002 pattern where you'd have high chlorophyll in the spring, declining in the summer, and then bumping up again in the fall. Since 2018, and probably earlier, there's just nothing in the spring, which is very consistent with some of the water chemistry I didn't manipulate her slide too much, but this was really the figure I wanted to show is that chlorophyll in the spring is just not a thing anymore in Lake Huron. Ewan Reavy at University of Minnesota Duluth, he's the guy who does a lot of the counting and investigating who's there in these samples of phytoplankton. 2017 at the top, 2022 at the bottom, and it's hard to see. The spring data is here in 2017, and then in 2022 the spring data here, but this really large bar, which indicates lots of phytoplankton, it's one sample. So it's not necessarily representative of the entire lake. But these ones here, compared to the offshore bars, indicates that spring is relatively devoid of phytoplankton. And the typical sort of systematic patterns you might expect to see in a oligotrophic lake, excuse me, where chlorophyll or phytoplankton abundance is higher closer to shore because proximity to nutrients coming in from the landscape, isn't very consistent. There are times when we see that in certain places, but then in other places it is not as apparent. This is a data again from a more targeted effort, which were a series of transects across North Channel, Georgian Bay, the main basin, and Saginaw Bay, looking to integrate lower trophic level indicators with larval fish recruitment. It's a little hard to see where the transects are, but there's one in Thessalon, Spanish River, French River, Parry Sound, Nottawasaga. That's Saugeen, Maitland River, Harbor Beach, Saginaw Bay, and Burnt Island at the top. Total phosphorus here is plotted just for the year of 2022, and it extends from early June to late August. And there are some similarities, but there are some differences. North Channel, for example, tends to be a little bit above the 5 microgram per liter objective at times. Saginaw Bay, obviously, it's more impacted. Georgian Bay, again, in parts where they were sampling. But South Main Basin, with the exception of early June, and the North Main Basin tends to be sort of hovering in that. But remember that these are not necessarily the whole basin, they are targeted locations. So there are places where total phosphorus is up around or above that objective. But what struck me out of this is that North Channel and Saginaw Bay were the places where it was most persistently elevated relative to other locations. And perhaps sort of confirming that, this is the same sites in 2022 with chlorophyll plotted on these. And again, North Channel and Saginaw Bay stand out as having higher algal abundance than, say, the Main Basin or Georgian Bay. This one is a different measure of wet biomass from colleagues at DFO, Warren Curry's group. And again, it's just for September, so it's later on in the year. The reference ranges here for the other lakes are from previous work, but you can see that Lake Huron samples from 2022, almost all of them fall well below Erie, Ontario, Michigan, and in some cases Lake Superior. There are some outliers, of course, in LH29, where there was a lot of Gleocapsa, which is a blue-green algae. It showed up in fishing nets and other things because it's got a very sticky mucus-like sheath, so it tends to cling to things. And then LH100 here had quite a bit of Microcystis and Alacosyra, which is a diatom. Again, just highlighting there are places where you can get outside the sort of overall main basin picture, but when you look across the sites, the phytoplankton abundance is still pretty low. Productivity, this is measured through carbon-14 uptake. The key thing here in all sizes of plankton, whether it's net plankton, nanoplankton, picoplankton, the spring productivity, which is blue, is almost always lower than that in summer. And and that corresponds to with the relative differences in the amount of phytoplankton. Patterns too, Saginaw Bay, much higher productivity than a lot of the main lake, Georgian Bay. Components, here you see North Channel actually a bit productive in the nanoplankton. I haven't got a sense of what this looks like in terms of if you smush them all together and made an overall picture, but certainly Saginaw Bay stands out as a productive place for plankton. Zooplankton, this is the only slide we've been managed to get at this point. A lot of the data is still being counted and identified. Some of the data is with partners who are currently under, we'll call it communication embargoes. So this is purely Thunder Bay and Saginaw Bay on the US side of things. 2017 is light green, 2022 is dark green, and there are some general patterns, but it's to me is hard to make out. The one point that was made by folks at NOAA is that the inner Saginaw Bay site had a lot of daphnia, which is why they had lots of zooplankton biomass in that particular year. There are some indications of other sites that it was higher in 2022 relative to 2027, but again, this is a single snapshot in time. Sort of the same with this, July and September. This is predatory cladocerans, Bithytrephes on the top, Sarcopagus on the bottom. What was interesting is Sarcopagus really only found in the North Channel, or sorry, the North Main Basin of Lake Huron, and was mostly absent in other places in July and completely absent from samples in September. Bithytrephes, variable amounts, of course, both in July and September, but this big one here in Saginaw Bay in September was that a response to the large amounts of zooplankton in Saginaw Bay earlier in the year? Don't know, could be just sporadic. Last one being mysids. Again, these samples are collected at night. They're there, primarily Georgian Bay, the North Channel, and the North Main Basin, not so much in the South Central and Saginaw Bay areas, but the number of stations that can be sampled successfully at night is relatively small, so we don't extrapolate too far with it. On the benthic side of things, I've split these into sort of the relative abundance, or relative proportion of things. Not much change in terms of the pattern overall. The largest contributors in terms of total numerical density are oligochaetes and zebra mussels and quagga mussels. Chironomids and spheroid clams holding pretty steady. The one notable change between 2017 and 2022 was we went from 125 total taxa to 126 due to the detection of an invasive gastropod at a number of sites. I've tried to summarize the changes in density in a table format here, where the blue arrows that go both ways basically indicate no change between the 2 years. Red arrows going down obviously indicates a reduction in density. In the brackets is sort of an indication of the fold change, and that's sort of an overall integrated number. The caveat being that in Dreissena density, sometimes in the shallows, so the 0 to 30 meter depth range, we might find more mussels there, but on the whole, in the other depth ranges, there's been a decline, and so in the main basin, the overall average is about 1.6-fold decline. Saginaw Bay saw about a 5-fold decline. Part of that was because in 2017, there were a large number of very young, recently settled mussels. Whether they would have survived all the way to adulthood is anybody's guess. The more disturbing part, I guess, is the continued loss of Diphorhea in Georgian Bay, not present in Saginaw, of course, and in the main basin, down about 3-fold from 2017. So the Diphorhea change is continuing to go down as Dreissenids, particularly the deeper zones, are coming down, is a bit concerning. The one note that they did make was that Diphorhea in the North Channel, the North Channel was where they found the highest number of diaphoria, but it's also the place where dry-seeded numbers are very low, which is sort of a good thing, I guess. The changes in the shallows, they're likely just fluctuations. The challenge we have right now, I think, is this a trajectory, or is this just interannual fluctuation of the population itself? Got 4 minutes, okay. I threw this in. It's from folks at the USGS. They were doing, this is the transect study referred to earlier. What was interesting here was the consistency between 2017 and 2022 of which types of fish they found in these locations. So differences obvious to present north to south and sort of east to west is what the dominant was, like rainbow smelt here in the north versus burbot and corrigonids in the south. But that pattern repeated 2017 and 2022. And my impression is that the density here, the larger circles indicate more larval fish relative to the smaller circles. So that was June, and this in the oblique toes, and this was the surface toes. As Ralph Hingley was the fellow who provided to me, he said what they were sort of encouraged is that they saw the most larval fish in places that were nominally more productive, and the species that were present were consistent. So there's some level of consistent agreement there. Closing up with, we are still pretty oligotrophic in Huron. There hasn't been much change in nutrient levels for a while. Spring diatom bloom has been missing for a long time. There is no evidence right now that it is returning in any way, shape, or form. Algal productivity is widely variable, space and time. There are local influences in some of these places. Don't have much to say in terms of zooplankton change as the data are still sort of coming in. And in the benthos, things are generally stable in terms of the types of organisms that are there, and there are some fluctuations in the density, but it's a little bit premature to say that things are going up or down in a consistent fashion at this point. The larval fish stuff, again, just closing with that, was consistent species composition and relative abundance between the two CSMI years. This was sort of the species composition and dominant species have some regional distinctions, which makes it a little harder, I think, for them to make interpretations as to whether there is a productivity bottleneck in terms of recruitment. But the densities that they did find were highest in North Channel, Saginaw Bay, and not so much in the main basin. I'm going to leave these as kind of an infographic thing. So Environment Canada, Climate Change Canada has now a program called EO Lake Watch, which is a remote sensing platform, which does have now near real-time processing and posting of information. This is just sort of the generic workflow. However, Lake Huron is going to be operationalized into this year, if not very shortly, which will allow people to basically observe how things are unfolding in a given year. Of course, I didn't put the web address on there, but if you type in EO Lake Watch in Google, it will direct you there. And that will be part, it's already opening and running for Lake Erie, Lake Ontario,